So welcome back to another episode of Explain Yourself. I am currently at EntSoc 17 and our first interview is with Lauren Diepenbrock. So say hi. She's so brave to be our first interview here at the conference, and I'm very excited to be speaking with you. So I like to start with your elevator pitch for your current research.
Ooh, I don't really have one.
All right.
No, I mean, so I work on an invasive plant pest, and so my goal is to figure out the ecology of this pest so we can rely less on pesticides. Right now, pesticides are the primary tool for management.
Mm-hmm. Okay, nice. Yeah, so that's something that's especially relevant to people and how people interface with nature, and that's a good, it's a good elevator speech, but I really like to make people do this thing. Are you a fan of XKCD, the comic?
I catch them every now and then.
Okay.
I am not good at following anything.
Okay, so they've developed this thing that's called the Simple Writer, and basically you're going to type in a sentence like, I study such and such. So simplify your elevator speech down to, this is basically the 100 simplest words in the English language, or 1,000 simplest words. So it's taking your elevator speech from sort of the up here level down to the everyman level.
That's good enough.
Alrighty. So Lauren's research statement is, I study a fly that did not live in most of the world until a few years ago. It eats food that we want to eat and is hard to control. I love that.
That
is
really
awesome.
hard to do.
But you did a great job. That's really great. Yeah, and I mean, while it's not very specific, anybody who hears that is going to have some idea of what you do. So I like that a lot. Yeah. That's great.
I totally have to go steal that.
That was beautiful.
Awesome.
Yeah. Okay, so now that we've done the elevator speech, you can sort of expand on that a little bit more and give us some more details, and you don't have to use the 1,000 simplest words now.
Right. Okay, so I'm a postdoc at North Carolina State, and I work on spotted-winged Drosophila, which is a fly. It is a drosophilid, so it's like a lot of the fruit flies that you get in your house that are super annoying. But this is actually more annoying because it is an invasive species. It's been in the U.S. since 2008 that we know of. It might have been here before that. And it uses a huge range of hosts. So it can use a bunch of stuff that we like to eat, which for the cultivated crops would be most berries.
Okay.
So blueberry, blackberry, raspberry, strawberry, cherry, the type of stuff, the soft, delicious stuff. And it lays its eggs in unripe and ripening fruit and ripe fruit.
On the plant.
Right. So it's like the stuff that is currently ripening and the stuff we want to pick right now. That's what that fly wants. It has no interest in the overripe, decomposing stuff that the Drosophilids that we know.
I was going to say, normally I think about fruit flies in my fruit bowl on my counter, and but these are in the fields.
It wants the same fruit we want. And so it's been hugely it's been a very expensive critter to control, and we don't really have full control of it. So the estimate is about $700 million annually for the U.S. for management for this thing. That includes management and how much fruit is lost.
Wow.
So for a picker or for a grower, if they have, say, a blueberry field and they pick all this blueberry fruit and it's in a cold truck going to get checked out by whoever's buying it, so the big companies that buy it and sell it at your grocery store.
Yeah.
If you find one larva in that truck, the entire truckload gets discarded.
Wow.
Yeah.
But I'm assuming that their larvae, their babies, are really, really small. So how do you find them?
The bigger, actually, I'm talking about this actually on Wednesday morning.
Awesome.
So the traditional way to do that is that you would do a salt flotation method, and so or some people use salt, some people use sugar. Basically, you make this hyper concentrated solution. You crush the berries a little bit, and you dump the berries in there. You stir it around. You let it sit for 15 seconds, and the larvae will kind of ooch out because all the salt water or sugar water is going into the berry. It's a really unhappy place to be. The larva comes out, and you can count larvae, but you can only see the big ones. So that's problematic because you can only tell if it's a third instar, which is a big guy that's if your fruit has a third instar, you're probably not going to pick it because the fruit's going to look like crap. And some people, if they're really good eyesight, can see the seconds. And so this is kind of what goes on normally at the farms. Blueberries also have the option of they have these really cool sorters that use lasers, optical scanners. They can tell off-color fruit. So if a basket fruit's slightly off color or slightly off texture, it is super cool. I've been in their packing rooms and I've watched it go. That's really neat.
Fancy.
Yeah.
That's really fancy.
So yeah, that's kind of the traditional way. And traditionally in the laboratory, we would rear out fruit. So we would take our fruit, put it in these little cups that have ventilation so that they don't. Basically, when they're eating the fruit, they're also fermenting the fruit. So they're basically getting themselves drunk and dying.
That's awesome.
So they ventilate, so everything kind of drains out except for the larvae.
Sure.
And then it takes like 10 days before we actually can rear out the adults. It's good for confirming who you have and getting an absolute number of who survived, but it takes a while, so it's not practical for like a grower or a field scout agent. So the newest method, and this paper just came out in September, and a couple of my colleagues, actually I have to give full credit to Steve Van Timmeren on this, because he was really the person who figured this out. The rest of us just helped write the paper and field validate for him. It's a paper that's in JIPM, so it is open access.
Okay, that's cool. So you can go and read the paper.
Yes, it is very easy. It is written for a broad audience, which is really nice because we want it to be applicable to a lot of people.
Yes.
So basically what he did is it's a salt extraction method, just like we were talking about, except instead of like trying to look at this clear liquid with like white squiggly things over who knows what background you're looking at, you can actually pour it through a specific size coffee filter, like those mesh coffee filters. And for most fruit, you can drain out all of the crud, you kind of rinse off your fruit, and all of the eggs through third instar can get caught in that. It doesn't work as well for blackberries and raspberries because they're super pulpy, and so they kind of just go to mush, and they kind of mush everywhere. And actually the biggest problem with that is just that the eggs get stuck in there. So the eggs get lodged in the fruit skin, but like blueberries, it comes right out. I haven't tested cherry because we don't have cherry in North Carolina. But from my understanding, it works really well with cherry also, but it's great. Like it saves so much time. Then you take this coffee filter, you put it under a stereo microscope, or if you're at a farm field, you just pop out a hand lens. You can see the eggs, you see the first, second, third instars, and anything past it, you probably didn't pick the fruit.
Very cool.
So yeah, it's a really simple method. It's beautiful. I can't believe I'm talking about it on Wednesday because it's totally his method. But he's talking about a different method in the same section. So
well, I'm going to put a link to this paper in the video description so you can go check it out since it's open access.
Yes, and there's some really beautiful images by Matt Bertone in there too, which is
very cool.
Really helpful for scientists as well as
Okay, so you said that this fruit fly isn't from North America, and I believe you said it was from Asia. How did it get here?
Well, we're pretty, pardon me, we're pretty sure that it came over on trade routes because we do a lot of international trade. And actually that's kind of the way that most invasive species move around is with our trade routes. And I mean, like in grad school, I worked on invasive lady beetle species. And so same deal, like you can follow the trade routes and you can see where things are moving. And we move stuff within the US, we move stuff up and down the Americas, we move stuff globally. It's really easy it's easy for somebody to hitchhike. And especially if it's something like a larva, a first instar larva or an egg of this thing, we think they probably came out on ornamental plants. Because they can use a broad range of ornamental plants in addition to the crop hosts. So there's a whole bunch of stuff, and we're still kind of figuring out all the stuff that they can use.
So any plant that I might get at like a nursery to put in my yard that happens to have fruits on it,
it could totally be infested.
Interesting.
It's ridiculous what these things can grow on. I mean, they've even found like larvae developing in mushrooms.
What?
Yeah, and there's a new paper coming out. I don't know if it's out already or if it's coming out soon. They can develop in like the compost from like cideries.
Oh my gosh.
It's crazy.
Flies are so like adaptable. That's frustrating.
It's a huge problem for managing them, which is why we mostly use pesticides. We have on the production side of things, we've started some practices like Heather Leach did some nice research looking at picking frequency, and so they pick more frequently than we even thought they needed to. My PI, Hannah Brack, did some work on cold management. So there's a whole cold storage management, cold chain management that happens to get the fruit from the field to the store and kill off most of those larvae, so you won't have a problem with them. Okay. And these are fixes for now too, and we're trying to look long term and to look at the ecology of the thing and how can we attack its ecology. So that's why I have a job.
All right. So are you looking at the pesticides, or are you looking at some sort of like non-chemical response?
So I started out actually working on pesticides, because that was what the original project was to figure out the optimal pesticide management regimes for blueberry and blackberry in the southeast, because we have a bit different climate than some of the areas where they've done previous research, which there hasn't been a lot because it hasn't been here that long. So there was some work in Michigan, and then there was some work done in Washington State on cherries. So it was kind of one of those things like we had an idea, but we really needed to quantify it. We really needed to put some numbers there that were useful for us, but also useful for the growers and realistic for the growers. So that's kind of the weird spot with doing this kind of research and extension, is that you do cool research, but it also has to be applicable for a grower, because otherwise, like, That's like we were talking about earlier with the so what question.
Exactly.
This is that research where I get to do super cool work, but what does it matter?
Yeah.
So I need to be able to say, okay, well, I'm doing this, I'm looking at the ecology, but bigger picture, long term, what does it matter? And so I've kind of picked up a couple things along the way and come up with a couple side projects that wound up being really useful. And our lab, or my PI, is the lead on a huge national project, so I've moved into a management position managing this specialty crops research initiative program, which is super cool because I get to work with scientists from all over the country, and they're brilliant and they know so many things that I don't know, and so I get to learn a lot from everybody else. Like when we have these big meetings, I'll organize the meetings and I'll organize an agenda, but I'll be like, I don't really, I get what you're doing, but I'm totally not the expert on this. So could you talk?
Yeah. And I completely agree with you. While I have nothing against pure science for the sake of science, I do think that the immediate utility of science like what we do makes it a lot easier to sort of convince people that it's worth it, and maybe makes our funding easier, hopefully, maybe. I don't know. And
I get to work with growers that are just really great people, and I learn a lot from them. So.
Yeah. So I think it's definitely more of an interactive science.
Which I like.
Yes.
It's more my personality.
Yeah. And thinking about interaction, tell us how it is to work with a large group of collaborators like that, because I know a lot of people think about scientists sort of on their own in the lab, but what is it like to be part of a cross the nation or multinational team?
Yeah. It was terrifying at first because I started in one position with this lab, and then we got this money and I got approached about it. And I was kind of oh my God, I don't know if I can do that. That's a lot of work. I didn't know if I was up for it. But I sat down and I really thought through, and I was like, wow, what a great opportunity. And hopefully if I start screwing things up, she'll tell me. So far we are finishing year 2. Yeah, we're in the end of year 2, getting all our data together, and planning our next big meeting in February. But it's been great. My PI put together a fantastic project team. We have people that if I ask them for something, I will get it from them. So the data, as far as managing data, getting data from people, we have just a really good team in place, and everybody works really well with each other. The hardest part is keeping everybody on task when we have big meetings. We have big, we use Zoom, so it's kind of like Skype. Okay, yeah. But a lot of extension people use that. So you can actually see each other face to face. You can share stuff on screen. It's a really nice interface. It's very simple, which I like. There's also the apps you can put on your phone if you don't want to have a full computer. But it's a nice way to actually see each other, to share the data, but keeping everybody to that time frame, because I don't like to waste time of my own or other people's. So when we schedule these calls, they're an hour and a half. And I will have every minute planned, and I will stop people if they go past their time. I'm like, next topic. And it's hard because some of them are really, really well-known scientists, and I'm just like, yo, postdoc over here, cutting you off.
So I guess that's something I wonder about. How do you sort of manage people so that you keep it an open atmosphere that everybody is excited to share, but you organize. Yeah. I'm
a big fan of agendas.
Okay.
I like to have very detailed agendas where people know going into it what the goals are of the meeting. And so the last big round, we just had a round of these top meetings. And I had quite literally, I was like, you have 5 minutes, you have 10 minutes. This was based on what I knew they had been working on and how much time they probably needed. And I touched base with the leaders for those different topic areas to see if it was reasonable and what kind of things they needed to talk about. So it's a lot of communicating and a lot of just making sure everybody knows what everybody else is doing and that we have the team going. And every year we have a big in-person meeting. And so this year we'll be meeting in Portland sometime in February.
All right, so you talked a lot about what you feel is the value of your research or why you're excited about it. Is there anything that you really dislike, or we could even use the word hate, about your research or any research project that you've done before?
I think I'm in a really lucky situation, to be completely honest. I work with a really good team. There are frustrating moments. Usually there's just not enough labor to get through everything. We work with a couple other research programs, which gets a little challenging, just juggling that much stuff going on, that many people and interactions. But really just the hardest part is there's so many cool ideas, and knowing when to say no.
Okay.
Knowing when to say no is the hardest thing, and you should try it out a little bit in grad school. I didn't, and it was a mistake. So I highly recommend like kind of thinking about what you can reasonably get done without killing yourself.
Okay, so you're talking about
life.
So you're talking about work-life balance and not being overwhelmed and
because I've hit that point quite a few times in the last few weeks, and I'm just like, what on earth am I doing?
That is a super valuable point. I think a lot of earlier career young professors and grad students are starting to recognize that. I think maybe the traditional idea of academia or the traditional way people approached academia was just like, this is your life now, and you have to take a break. So don't do research all the time.
I mean, it's tempting because there's so much cool stuff, and like if you don't get in on it, you might miss out. But at the same point, you just have to realize that you can't do everything. You do need to sleep. Yeah,
and I think like if you tried to do everything, it wouldn't be as good.
No, it would be crap.
Yeah.
And then you don't want to be that scientist who just puts out crap.
No. Yeah.
Like, you want your stuff to be quality.
Yes. All right. Well, so we're sort of working our way backwards here, but I know that you're a postdoc, so that means you also did a doctoral program. Did you also do a master's?
I actually did 2 masters because I'm crazy.
Awesome. Okay. So tell us a little bit about like your journey from like undergrad through grad school and like why you decided to go to grad school. And
so this is I took a very twisted road to get to where I'm at. As an undergraduate, I got to do research. So I started undergrad thinking I was either going to go to med or vet school.
Okay.
So I went to University of Missouri. It's a big state college. It has a med school and vet school. Great pre-programs for both, and reasonable price. Because that totally factored in.
Yeah.
Because my sister's older and she went to a very expensive college, and I watched that. I'm like, no, not happening. Because I'm going to go to like grad school, med school, or vet school. I don't know which one, but I know it's going to be something.
Yeah.
So I got in there, and about my second year, I started realizing I did not want to go to medical school. I did not want to go to veterinary school because I didn't really like my peers that much. I was like, they're not very nice. They will step on you to get where they want to be, and I didn't want to be around those people for the rest of my life. And so I saw this lab tech job for a summer, and I just called the person, went up there, met with them, and I wound up working on C. elegans with Don Riddle, who apparently is like really well known for C. elegans and his work with genetics of C. elegans.
So that's it's a model organism used for genetics research, and it's a nematode?
It's a nematode.
It's a nematode, yes.
It's very commonly used for molecular work and genetic work because you can manipulate its background, and there's so many strains that have been manipulated, and they're very easy to get a hold of. Huge amount of people that work on it. And I just started out washing dishes and making agar plates and keeping colonies alive. And then end of my junior year, they said, hey, do you want to do research? And so I wrote like a small grant with the help of the lab manager and the professor, and it was internal grants. They have a life sciences undergraduate research opportunities program there, and it's awesome. And I was accepted for that summer and for my senior year.
Yeah.
So I was able to do this whole research program, develop a project, do the project. It totally flopped in the end. Like the data looked great, and then we decided to get a couple more runs in before we tried to write it up, and then it all just went nothing. Like nothing was consistent. So I learned to handle failure at a very academically young age.
That's super important, though. Yeah.
I was really frustrated, and I was like, "What? Seriously, what? Come on!" But then from that work, I'd actually been recruited to do a PhD in biomedical research. And I made it about 6 months, and I realized I absolutely hated it. So I started this program. I met just some of the most interesting people I've ever met in my life, because a huge diversity of backgrounds, people from all over the world in this program. We had 20 students in that class, and I still talk to most of them, which is kind of cool.
Awesome.
But I just hated it. I was like, this is not what I want my life to be. And so as it turns out, I was in New York, upstate New York, Syracuse area, and Syracuse has a really good education program. And their science ed master's program at the time, it was the second best in the country. So I thought, maybe it'd be fun to teach. So I applied, I got in.
Yeah.
I did my master's in science ed, and then I went back to St. Louis where my family is, because free rent. And I taught high school in the biggest high school in St. Louis for 2.5 years.
Wow.
Freshman chem, Gen Bio 1 and 2, and AP Bio.
Is that what people would call like an inner city population that you were working with?
Yes, for the most part.
Cool.
It was a really interesting mix of students because you had kind of the two extremes, and the school didn't really offer a whole lot as far as like resources for teachers. It's kind of one of those things I look back on. Like, I'm glad I did it because I learned a lot about teaching. I saw a totally different set of life from what I knew. And actually, my students, when I left to go back to school, they're they made me get on Facebook so they could keep in touch. And so I've kept in touch with a lot of my former students.
Great.
And it's really fun to see where some of them have wound up. Like I have a student that recently finished her PhD in biomed, a student that's a pharmacist, a student that runs a YMCA youth education program. Like they're doing cool things. So it's nice to see where they wind up. But yeah, then from there I was bored. Teaching's fun to a point, but mentally I had like nothing. I was just like, my brain is just not functioning. So I started applying to grad schools, and I got a phone call one day from Walter Schinkel at Florida State, like the fire ant guy.
Yeah.
Literally, he wrote the book, The Fire Ants. And we wound up talking for a while. He had seen my application packet, and he was like, all right, I'm going to offer you an opportunity to come down here for graduate school. And he always starts his students off with a master's, the option for a PhD. And so I got down there, and it was really cool because he said, you can do anything you want as long as it's on ants. And I too had to TA through that. So I did a lot of teaching.
But anything on ants is such a huge, oh wow.
It was really fun. So it was cool because I tagged along with him. I tagged along with the postdoc, Josh King, who's now faculty at UCF, the University of Central Florida. And when I was working with Josh one day, we were digging up these ants to send off to a developmental lab, and they were trapjaw ants.
Oh.
I'd never heard of a trapjaw ant. They were really cool. And I had all these questions, and Josh just looks at me, he's like, I think you found your project.
Yay!
Because it was all natural history that we didn't know.
Yeah.
And so for 2 years, I dug up ants, and I studied their seasonal natural history, and that was my master's. It was really fun, and it's kind of cool to see all the projects that have come since on trapjaws, and like the different trapjaws and some of their mechanisms and how they do what they do, and I'm just like, this is so fun.
That's
They're just
amazing.
a cool ant. Yeah, and while I was there, I was able to kind of hone in on what really interested me, because there's a big ecology program. And so I knew I didn't want to stay for my PhD because it wasn't exactly, I really wanted to go like entomology, like insect ecology.
Okay.
And so I started applying to programs, and I actually got back in touch with a professor I had for an undergrad course. And I was like, so at the time I actually met my now husband the day I accepted at Florida State. So we long distance dated for 2 years, and I was hoping to try to get back to somewhere nearby so we could actually see. So anyways, I got in touch with this professor, and I was like, this is what I want to do. Do you have anybody? Because the website wasn't very good. And she was like, by chance, we have this like new professor. And so I emailed the new professor, who is Debbie Finke, and she's awesome for insect ecology. I emailed her. We got along really well via email, so I came up for an interview, and she offered me to come do my PhD. Which was cool because I was her second student, so I got to like develop a lot of my own stuff, like find my own research sites. It wasn't already an established system, so I had to figure that all out in Tallgrass Prairie.
I was going to say, that's both fantastic and scary to be the first one to like establish a research site.
Yeah, because there's a lot of questions that you're like, when you go into your exams, like, so why'd you pick this? Did you control for this? And
Yeah.
like, yes, I have this incredible insect ecologist as a mentor, and so she already asked me all these questions. So yeah, I studied lady beetle ecology in Tallgrass Prairies for grad school, and in part of that, looking at the impact of invasives, so the 7-spot and Harmoniax viridis, the multicolored Asian lady beetle, on native communities in these tallgrass habitats. So we looked at native prairie, so it's always been prairie. Those are really hard to find, by the way.
Yes, they are. We've talked about that.
Yeah. And then restored prairie sites, which are slightly easier to find.
Yes.
And then tallgrass agriculture, so fescue.
Sure.
So you had an ag control. And so we were looking at the lady beetle communities and a couple of resource factors, and then the impacts of the non-natives.
Nice.
So, yeah. And then I got really interested in invasive species ecology. I was like, well, that's really cool. I wonder how this works in an ag setting. And it turns out, like, if you ever want to fund a lab, you should have an ag bend to your work.
For sure. Yes.
Yeah, and so then like this, when I was graduating, there were just a handful of positions. For some reason, there were very few postdoc positions that year in anything I was interested in. And so this one opened up, and I was like, wow, that sounds super cool. I know nothing about pesticides, but it's an invasive fly. It hasn't been here that long. There's probably lots of stuff I can do. And so the professor actually wanted somebody to start before I was technically defended. So I emailed her, and I introduced to myself and who I am, what I was interested in, and I was like, I'm really interested in this position, but you want somebody to start on this date. This is the day I defend. Are you flexible? And she emailed me back a little bit later, and she was like, absolutely, send me your application.
Yeah.
Yeah. And so I interviewed, and I actually interviewed in that like gap. So you submit your dissertation, you usually have like about a 2-week gap between that and your defense.
Yeah.
So I went for my interview during that period, which everybody thought was crazy. But I was like, what else am I going to do? It's 2 weeks. It's my butt for 2 weeks.
Yeah.
So I went down there, and it was basically my practice run of my defense. And I loved it, and I was just kind of waiting. An hour before my defense, I got a job offer, so I walked into my defense like, I got a job.
Yes! That's so awesome.
Yeah, I lucked out. It's been a really good fit.
That's great.
The lab's been it's been a good opportunity because my advisors also had 2 children the time I've been there, so I've gotten to step up into an advising capacity a little bit and work with the students more. I've gotten to do some international travel, gotten to just have a whole bunch of cool experiences that I probably would never have had otherwise.
That's awesome.
So,
And I think your story is a good example that like, just ask, right? Like put yourself out there, don't be afraid. Like if it's at all interesting to you, just go for it, because like your energy and your passion are going to be what pushes you into the things that you want. So
and
I like that.
I figure at this point, I know enough scientists to know we're all a little awkward.
So we're backing up a little bit further, and you told us that you wanted to go to school for science, but not anybody else in your family is a scientist.
Yes.
So where did that inspiration come from? Why did you want to be a scientist in the first place?
Oh, honestly, it was the only thing I was interested in. So I mean, I was saying, I grew up in a small town.
Okay.
And there was a lot of woods to play in, so there was a lot of things to play with.
Yes.
And I didn't really recognize it as science per se at the time. I mean, there's like bones of a rattlesnake that you played with, or like a frog that you found dead in the house. And so I was that kid that was interested in all those things, like how things worked. And so my parents recognized early that I really liked science. And I was really lucky because they bought me a microscope, like a kitty microscope.
Yes, I love those.
And they bought me a whole bunch of like chemistry. So again, we lived in Florida, so all of my science stuff had to stay in the garage. So I had all these chemistry kits. They had made me this little lab bench, and so I had all this stuff. Most kids don't get this, but it was really fun. And I could bring in the things from the woods that I found and look at them in the microscope and play like with my little chemistry sets. And I was obsessed with Mr. Wizard. Like I had every single Mr. Wizard book ever, and that might be like way too long ago for you to even know that name.
I know who that is. Yes,
Mr. Wizard was my friend.
That's such a fantastic opportunity. I'm glad they encouraged you to do that.
I mean, I'm lucky. Like my parents actually really encouraged us to follow things we were interested in, and so music and science are like the only 2 things I've ever been good at. I didn't know, because I didn't have anybody else in science, and I really didn't have anybody outside of my science teachers growing up. I didn't know what other opportunities were out there, and so like I tend to take all the opportunities to go out and talk with student groups when I can to say, hey, there's all these cool opportunities of things you can do.
Yeah.
Because I didn't know. I thought you had to go to med or vet school if you were good at science. Like that's what, how else are you going to know that?
Yeah. Is there something that you wish you had known, like about grad school or about like becoming your own researcher? Like you wish you had known that before you started? Like if you could go back in time and tell younger Lauren?
See, I learned things the hard way a lot of the times. Definitely that whole saying no thing that we talked about. I was really bad about that in grad school, and I was like, yeah, sure, I'll do that. And I wound up taking on a lot of responsibility, which in the end, a lot of it turned out to be good, but sometimes it was more than was reasonable for a graduate student at the time. And I roped in my free labor of my spouse for things, and yeah. I would highly recommend that whole take a little time for yourself. I suck at it. It's something I'm still working at.
And also, what do you think is the thing that you still need to work on the most as a researcher? Is it that, or do you have something else?
Probably learning to hold my tongue at times. I'm really bad at it.
Oh man.
It's something that I've never been good at, and it was when I was teaching high school one of my biggest problems because I didn't see eye to eye with a lot of administrators. So yeah, learning when to say certain things and not say certain things.
Or how to say them.
Yeah, I'm not very tactful.
I can totally sympathize with that.
Yeah, it's not something I was ever really taught. It's something I'm working on.
So are you just giving a talk here, or you're like moderating a panel and you're also judging the student talks? So like, how do you sort of approach coming to conferences, and like, is this the only conference you come to, or do you like to go to like a lot of different kinds of conference?
So it always depends. This is one I consistently come to.
Okay.
And normally I'm much more prepared than this year. This year I am extremely unprepared. One of my 2 talks is not done. One talk I finished last night. Yeah, but in all fairness, I've been doing a lot of other things this year. I like to have everything organized before I get here. Like I kind of like to come in with a basic plan of attack of what talks I want to go to, what sessions I want to go to, and I usually have all my talks ready beforehand. But not this year. So I did look at the schedule ahead of time. So yeah, this morning I judged a session, and then this afternoon I have a couple meetings and a couple mixers and other social things to do. And yeah, tomorrow looks a little crazy, but I gotta find some time to finish the other talk.
Yeah.
I do try to go to the branch meetings when possible. I really like them. I think as a student it's getting harder to go to those because the funding's less and less, but I think as a student it's actually the best meeting you can go to. Like there's a lot of great stuff here for students, but it's such a big meeting. So the branch meetings allow students to have more one-on-one time and more quality interactions with people.
That would be nice.
Yeah.
But yeah, there's very limited funding to travel.
Yeah, when I was a grad student, we always had money to go to North Central Branch. But I always played Linnaean games, so that paid for my trip. So, and if you haven't seen Linnaean games, totally go check it out. It's awesome. It's like Bug Nerd Bowl. It's fantastic. I love it.
That sounds like fun.
Yeah, all sorts of bug trivia. Yeah, and it's really fun because students have to go and compete at the branch level, and then you compete at the national level if you make it past the branch, or if the top 2 teams make the branch come. So it's really fun to watch, and it's just amazing the knowledge these people have. Because I never felt like I knew that much, but I was always like, I can answer a couple questions. Yeah. So I just try to come in prepared as much as you can. And as far as the other meetings, it's kind of whatever I get invited to talk at, because I'm on a budget. So if they're going to pay me to come, like my trip to Australia a couple weeks ago, I was like, yeah, but I don't have no money, so can you pay for it? So I got to go give this talk at the Australian Entomological Society meeting, which was it was a neat meeting. It was a totally different setup than what I was used to, so it's cool to see those. And the interactions were great. I met a lot of people who have similar interests, and they're really worried about this fly that I work on. So I had a lot of good interactions there. And I wind up going to a lot of grower meetings and talking to a lot of commodity crop meetings.
Sure. Yeah.
So like I'll be at the strawberry meeting in January in New Orleans, and right after that I'll be at a meeting in Savannah. It's like a regional grower meeting.
And that's awesome because that means you're actually communicating your science to the people who need it.
Yeah. It's just really fun because they have different interests and needs and questions, and it's a different approach to presenting your data. So I actually find those talks the most challenging talks to put together, because I could talk at the level of most of the people that write the literature, but that's not the level that I need to be communicating it to be effective. So I'm seeing this more and more when I write my talks for these meetings, is I'm actually trying to take these talks to be more approachable too. Because my spouse is with me at this meeting. He's not a scientist, so he sat through some talks this morning and was like, I have no idea what they talked about. And so I've been trying really hard to not do that.
And that's why we do this, because I think that it is important for everyone to talk more like a normal person and less like a robot or a scientist or whatever.
Yeah, and we're not really trained to do that. And I understand, like, looking at the judging forms, like, oh yeah, well that's kind of why we talk this way, because this is what we're taught to talk like. So maybe we need to think about revamping some of that. But yeah, it's really important to think about your audience when you're putting these talks together and make sure it is appropriate.
Yeah.
So
yeah. And final question to wrap up. So what is next for you?
Hopefully a faculty position. I mean, I've been interviewing. I'm waiting to hear back on my most recent interview, so we'll see. I've got at least another year that I can be a postdoc if need be. NC State has a max out of 5 years as a postdoc, which is fine. Like, I would love to have my own lab.
It's better than being trapped in a postdoc. I know there are lots of places where you could just get stuck there.
Bounce postdoc to postdoc to postdoc. Like, I was talking to a guy earlier who's quite literally, he's been 10 years of bouncing, and I'm just like, financially, that's not an option. It's just not an option. Okay. I have student loans to pay. Yeah.
Well, I wish you lots of luck with your faculty position search. I know those things are becoming less and less available, but you obviously are very passionate about this and totally deserve a good position.
Let's hope.
And I so much appreciate you coming and speaking to me and allowing my audience to learn more about, how entomologists do things. So thank you so much. If you liked this video, don't forget to like it. If you didn't like this video, please share it with someone who would. And if you'd like to support the Roving Naturalist, don't forget to hit the subscribe button and the bell icon so you can get notifications when I publish new videos. You can also follow me on Facebook and Twitter, and I'll see you next time.